As a bit of a primer, we'll do a brief circuit of the cell to see how we're going to explore how virus infection and release from a cell takes us through the different parts of the cell machinery. First off, a virus has to get into a cell. Hence, we'll have to look at the viral cell surface, the proteins on its surface, and how it's recognized by the cell, and then how the cell responds in the process of receptor-mediated endocytosis. This is the process by which most cells import material into their cytoplasm. Next, we're going to look at how they get out of the endosome, and how then the components of the virus, most importantly the RNA, gets out of the virus and into the nucleus where copies are made and information for new proteins is generated. So we have to look at how endosomes are regulated, and then how molecules in the cell, like RNA, are targeted to different parts of the cell. Then we're going to look at how the RNA, once it's turned into protein, how is protein synthesis regulated, and also when proteins develop, how do they know what part of the cell to go to? How are they packaged? How are they targeted? How are they shipped? We have to explore all these aspects of cell biology in order to understand how the virus gets through the cell and gets its particles to a place in the cell where it can be assembled. And finally, we look at the formation of the new virus particle, that is, self-assembly, budding, and release. It's a marvelous aspect of viral particle and many aspects of cell biology where molecules have a natural pattern of interaction with each other that helps them to self-assemble into complicated structures. So in fact, the viral particle is self-assembled simply based on the properties of association between the constituent molecules. Okay, so a virus is made out of the major classes of macromolecules. It does an interesting job of getting into cells and mucking about. So is a virus a living thing? Well, I'd like you to vote and let us know in the next class. And if you want to explore a bit further, here's a URL where you can see how other people register their opinion. As I mentioned, I'm using the influenza virus because it's a terrific model to integrate the basic concepts of molecular cell biology. But secondarily, the influenza virus has had a major impact on human history, and you've probably had some personal experience with it, so that makes it a more relevant model to review as we go into this course. So, there are many strains of viruses out there, which is why we have different epidemics. The major epidemic of historical importance is the Spanish flu, which we term the H1N1 virus. The current bird flu, Asian bird flu, is actually an H5N1 virus. So in the next class, September 11th, I'm asking you to tell me which the H and N numbering system is based upon. So we've had recorded flu history that dates back to 412 BC when it was first described by Hippocrates. Not until the late 1800s was it known to be associated with domestic birds, as it was called fowl plague. And then in 1918, there's the major world epidemic of the Spanish flu, where almost a quarter of the world population caught the flu and over 20 million people died. There have been various little epidemics since then, and now in 1999, the H5N1 bird flu virus emerged in Asia. So what does it mean if bird species are a reservoir for the influenza virus? It means that birds are constantly carrying influenza viruses where they evolve over time to evade their immune systems. They can change there then in ways that make them more adaptable to invade other species, including human ones. So particularly in situations where bird species and humans are in close proximity, such as in farms or intensive farming, the possibility for mixing of the avian virus and the human virus is possible. This happens when both viruses, from one common to humans and one common to birds, infects the same cells in a human being. The genetic information can then reassort itself to create a novel form of virus, one that humans or birds have never seen before, and is particularly potent because our immune systems don't know how to handle it or recognize it. Okay, so you catch the flu. Why does it have to be a bad thing? We have plenty of energy in our bodies, we can spare a little for the flu virus. Well, the problem is that when the flu virus infects a cell, it pretty well takes over all of the machinery and devotes the entire cell to making more viruses. The result is the cell ultimately dies. In our case, because we inhale the virus and our respiratory tract is most vulnerable to this kind of aerosol attack, the virus will ultimately end up killing a lot of the respiratory epithelium. Then we can't properly regulate our respiratory system. So what happens next? I'm asking you to put the pieces together from the rest of what you hear in today's lecture. So I will ask at the beginning of the next class, how can you die from influenza? I hope you don't meet the influenza virus in residence this year. Let me recount for you what happened in the barracks of the American Army after the First World War. Camp Devens is near Boston and has about 50,000 men, or did have before this epidemic broke loose. This epidemic started about 4 weeks ago and has developed so rapidly that camp is demoralized and all ordinary work is held up until it has passed. All assemblages of soldiers is taboo. These men start with what appears to be an ordinary attack of la grippe, or influenza, and brought to the hospital, they very rapidly develop the most vicious type of pneumonia that has ever been seen. 2 hours after admission, they have mahogany spots over the cheekbones, and a few hours later you can begin to see cyanosis extending from their ears and spreading all over their face, until it is hard to distinguish the colored men from the white. It is only a matter of a few hours then until death comes, and it is simply a struggle for air until they suffocate. It is horrible. One can stand it to see 1, 2, or 20 men die, but to see these poor devils dropping like flies sort of gets on your nerves. We have been averaging about 100 deaths per day, and it's still keeping it up. There is no doubt in my mind that there's a new mixed infection here, but what, I don't know.